Hello everyone and welcome to the Dichotomous Ocean Dinoflagellates presented by Alexi Turner. Dinoflagellates are part of the protists kingdom. My old biology professor likes to say this kingdom is akin to the junk drawer of taxonomy. They're a collection of organisms that do not fit into any of the other domain or kingdoms already classified. Most have both plant and animal characteristics. They can be autotrophic, heterotrophic, or even parasitic. Most protists are unicellular, however, there are colonial and multicellular examples as well. Dinoflagellates are a group of unicellular aquatic organisms that have multiple functions in our ecosystem. They are incredibly diverse in their appearance, as seen on the title slide, but they all have a transverse groove, which you can see here, and two flagella, which some is seen here. Once again, they can be photosynthetic, heterotrophic, or even toxic. And shown here is a micrograph of the genus Gonolox, which are known to cause red tides. Today I'll be focusing on two main functions. This includes the symbiotic relationship with cnidarians and the production of red tides, which produces the neurotoxin. Mutualistic dinoflagellates are fairly studied and well known. Dinoflagellate produces sugars via photosynthesis for the corals for the maintenance, growth, and calcium carbonate deposition needed, and in return the corals provide nitrogen to the dinoflagellates. Photosynthetic species that live in corals are called zooxanthellae. They also provide the corals the characteristic bright colors, as you can see here in this image. The family Symbiodoniaceae are the main mutualistic dinoflagellates that live in corals. And reef-building corals, such as stony corals, as seen here, in shallow, nutrient-poor waters are the ideal host for these relationships. This intimate mutualism is the basis for the existence of all shallow water coral reefs on Earth. A study done in 2012 looked at evidence that glucose is the metabolite transferred in cnidarian dinoflagellate symbiosis. This is the cnidarian they used, this is the dinoflagellate they used. Carbon-13 glucose fragments could be detected in light incubation, as seen in graphs A1 and A2. And as expected, there was no significant labeling of glucose observed in dark incubation in A3, in the absence of dinoflagellates, which is in D over here, or using carbon-12 labeled glucose in A4. One detrimental issue is coral bleaching due to the rising sea temperatures that's killing off these dinoflagellates. The average sea surface temperature for January 2025 was 20 degrees Celsius, which is the second highest value on record for January, and it's projected to increase. With these increasing global temperatures, the coral bleaching risk increased immensely, as you can see in this map below. If these dinoflagellates keep dying, the yellow-orange sections here will soon turn red and purple. While this family of dinoflagellates is incredibly beneficial, a different species is deadly. The red tide is a phenomenon where normal marine dinoflagellates, Caronia brevis, grow rapidly and overproduce neurotoxins that kill and contaminate fish, animals, and the air. The most common occurrences are typically along the southwest Florida coast, which is seen in these pictures here. They get their red color from the dinoflagellate pigmentation. Brevetoxins are neurotoxins produced by this dinoflagellate, which cause a massive fish die-off, marine mammal killings, and many other impacts of the ecosystem. Humans are also impacted through the consumption of contaminated shellfish, which is known as neurotoxic shellfish poisoning. The last terrific red tide in Florida was in 2021, which resulted in 600 tons of dead fish and marine life and 36% decrease in marine recreational sales. So what causes these organisms to proliferate and grow out of control? Well, scientists aren't really sure. According to the Texas Parks and Wildlife, a certain unknown combination of temperature, salinity, and nutrients is needed. A novel idea is that viruses could impact the blooms. A study done in March 2025 discovered ssRNA viruses associated with K. brevis. This is important because factors like viral lysis could result in boom decline. Understanding and studying these unique organisms could have potential beneficial impacts on our ocean ecosystem. Scientists could research symbiotic dinoflagellates that live in warmer climates and implement those proteins into the family Symbiodoniaceae that are being bleached. In addition, scientists could find a way to reduce the red tides if they understood the conditions needed to proliferate these blooms. In conclusion, it's time for scientists to better organize that horrific junk drawer of protists. Here are my references, and thank you for listening.